smells like a garbage truck. No, I'm just kidding.
All right, so as we're waiting on people to roll in, just a couple of quick things. I know we covered some kind of logistical stuff this morning in lecture regarding your final exams and stuff like that. But you guys are better off than my morning lab because you are less than a week from being done with the class. At this moment, a week from this moment, you will be finished with the class. How exciting is that? So neat. Huh. One of the things that I'll be doing in the next couple of days is I'll be posting my office hours schedule for next week because it will be different than normal because of final exams. So I'll be posting that soon in Canvas as well as on my office door. But as far as logistical stuff goes, one of the things that I thought would be kind of fun is if we tried over the next week, because you guys are going to have a few more days, tried over the next week, and I know it's not a lot of time, but you guys will notice that I brought a food drive box into the room. So as many of you guys know, not everybody can afford to buy groceries, and with the holidays that can be especially hard. So I thought it would be fun if we saw as a class how much food we could actually get donated to the 50B class. Not just you guys, but all of the 50B. And I thought it would be extra fun if we included 50A in it. So there's a box next door in 50A. And I thought it would be fun to have a competition to see who could actually bring in and get the most food donated for the holidays. So this is here. If you guys have non-perishable food that you can bring in, and if you have the means to donate, you can just bring it in and put it in this box. And I think it would be fun to see how much we have. It looks like somebody's already used it for the trash can. I'll clean that up today. It's for non-perishable food only, though. So please, if you have the means, if you have some at home, even if it's just a can of something, bring it in and put it in that box, and we'll see how much food we can get donated over the next week. Because we just came off of Thanksgiving, and it's important to realize, to remember at this time of the year that not everybody is as fortunate as everybody else. So it's nice to be able to make an impact where we can. So I thought it would be fun if we could compete with 50A on that and see what they can do. They have more students, but we're cooler, so I think we can do it. All right, so we'll see what we can do. So that's the only kind of lab-specific announcement I have is let's try to get that box filled over the next couple of days. All right, so just a real quick recap of where we were at in the lecture this morning. We talked a little bit about kind of the human life cycle. We talked about the fact that we sort of alternate between this diploid phase of our life cycle, and then we produce these haploid cells, these gametes, these sperm and these eggs, which we need to, of course, combine with other gametes to make new viable individuals. And that's what we do as sexually reproducing organisms. So today what I wanted to do in lab is I wanted to start to look at the reproductive tract of both the male and the female anatomy. Took us a little bit longer this morning than I think I maybe planned on, so we only got through the male reproductive tract this morning. So on Friday in my morning lab at least, we'll need to finish the female, which we should easily be able to do. And give you guys time to review for the final. But we moved a little bit slower this morning than I sort of anticipated, which is totally fine. So remember that both male and female reproductive tracts need an organ to make the gametes in. That would be, in the case of the male, the testis. And then we need a duct system to get those gametes out of the body. So that's kind of what we want to look at. So I wanted to kind of go through some of the reproductive tract in a little bit more detail than we got to in lecture this morning. Show you a couple of models. I'll let you guys So, as we were saying this morning in lecture, the testes are housed in the scrotum. And one of the things that we said is that, there are some species of mammals that have this ability to sort of internalize the testes between mating seasons to help to protect them. But as primates, we really don't have a defined mating season. So as a result, we don't have that ability to move the testes into the body cavity. And the scrotum, as I mentioned this morning, is really kind of a fairly boring sack of skin is really all that it is. But there are a couple of interesting features about it that I wanted to mention. Back to this. So if we open up the scrotum to reveal the testes, one of the things that we'll see is that there are multiple layers of connective tissue. The only one that you're actually being asked to learn is the tunica albuginea, which is the white layer of connective tissue directly covering the testes. But there are several other layers. And in those layers are a couple of muscles that I think are worth pointing out. So here we've got a couple of different layers of dissection in this diagram. And I know that this diagram is a little bit dated, but it's actually a pretty good one. But what we see is right here in the skin of the scrotum, you'll notice that they have that sort of red line sort of outline. That red line is a muscle that is embedded in the skin of the scrotum called the dartos muscle. And I'll show it to you on some models as well here in just a couple of minutes. It's a really, really thin muscle. It's no thicker than a piece of tissue paper. But what it does is it can essentially control the skin of the scrotum to cause it to either contract or relax, largely in relationship to temperature, right? So we said this morning that sperm have to be produced at a lower than normal body temperature. Anywhere from 2 to 4 degrees below body temperature is the ideal temperature for sperm production. So, if things are really, really cold outside of the body, what we can do is that dartos muscle can contract and actually contract the scrotum and pull the testes a little bit closer to the body cavity. Whereas when things are really, really warm, the dartos muscle can relax and allow the testes to hang a little bit farther from the body cavity. And what that does is it creates a little bit of a temperature control system for the testes. Okay, so that dartos muscle is a really, really thin muscle that's embedded in the skin of the scrotum. And we'll be able to see that on a couple of our models today as we kind of go through this. If we go past the scrotum and we look at the connective tissue surrounding the testes themselves, again, there are going to be multiple layers. But in those layers of connective tissue directly surrounding the testes are another muscle called the cremaster muscle. And we see those fibers of the cremaster muscle shown around this testis here in red. The cremaster muscle essentially does partially the same thing that the dartos does. It works to help to regulate the temperature of the testes. But remember that the dartos is only around the skin of the scrotum, so it can only kind of control that. The cremaster muscle, being around the testis itself, can actually pull the testis up a little bit, right? So it has the ability to do that. So those two muscles kind of work in unison with each other to pull the testes up in regards to a lower temperature, and then they relax to allow the testes to hang lower from the body cavity in terms of an increase in temperature. The cremaster also, to some degree, serves a protective reflex as well. This part of the body is particularly sensitive to touch. And when it is touched, the cremaster muscle will actually reflexively contract and pull the testes closer to the body cavity in an attempt to help to protect them. It's called the cremasteric reflex. You can actually trigger it. All you have to do is touch the inside of your thigh, and you can actually trigger the cremasteric reflex. watch the testes whoop, go closer to the body cavity when that happens. It's kind of an interesting thing. So I don't know if there's a video. There's probably a video, but I'm not going to go to Google for that. Not on the school computer. But there's probably a video of it online. But you can actually, if you have the anatomy available to you, you can actually watch it in real time just by touching the inside of the thigh. It's kind of an interesting reflex, but it's called the cremasteric reflex. And it's just a protective reflex that allows the testes to be pulled closer to the body cavity, hopefully protecting them from damage. So 2 interesting muscles that are shown here, and the models do a pretty good job of showing them to you, and I'll show you here in just a couple of seconds. So that's kind of 2 muscles that I wanted to point out. We will come back to the anatomy of the testes in a little while. I want to revisit those. We do have a microscope slide of the testes to look at. But suffice it to say for right now, excuse me, the testes are really just comprised of these seminiferous tubules, miles of seminiferous tubules that are just condensed into each individual testis. They're found between pretty thick layers of connective tissue called the tunica albuginea. And then as we said this morning in lecture, those seminiferous tubules kind of coalesce and become the epididymis, and then the epididymis becomes the vas deferens, which leads out of the scrotum and into the abdominal pelvic cavity. The vas deferens is sort of bundled with blood vessels and nerves in this structure called the spermatic cord, and we'll be able to see that on this model here in just a couple of seconds. Right? Anyway, we'll take a look at the microscopic anatomy of the testes a little bit later on today, but let's take a look at some of the gross anatomy here of the male reproductive tract. One thing worth noting is that we have several models available that all of them, I think, have different strengths and different weaknesses in terms of showing different things. So it's definitely now, like more than almost ever in the semester, really worthwhile to spend time looking at all of the models that we have, making sure that we have good labels on everything and stuff like that, and you can study with at home. I'm going to start with this one, and then we can kind of go from there. But this is a good one to start with. It shows everything pretty well. It's a good 3-dimensional model of the male reproductive tract. So we'll go ahead and start with this. All right. So obviously this thing is in various phases of dissection. The skin has obviously been removed from the front of the scrotum and the penis and stuff like that. We'll get into the penis eventually. We'll see the back of it from sort of a 3-dimensional way as well. But it does come apart to become mid-sagittal as well. So it's a really cool model in a lot of regards. So let's zoom in and let's go ahead and get started taking a look at this. We'll start with the scrotum and the testes, and then we'll work our way out from there. So let me actually remove this so that it isn't in our way while we chat. You have the skin of the scrotum right here, of course, and then you have all of that connective tissue covering the testis. One of the things that's immediately obvious is the network of red lines that you see. This is showing the cremaster muscle. This is that muscle that directly surrounds the testis and can then contract and pull the testis upward in regards to temperature or touch or other stimuli. The other side of the model, you'll notice right here. The other side of the model, you see here you have that real thin red line. That's that dartos muscle that's embedded within the skin of the scrotum itself. That essentially serves the same purpose, but it's the one that is directly responsible for adjusting the tension on the skin of the scrotum. So we see that right here. That said, though, this side of the model is more dissected away. All that connective tissue has been removed, so we can see the testis itself. We can see the epididymis right here, and then we see that vas deferens sort of leading away, and then it becomes packaged with blood vessels and nerves in the spermatic cord kind of up here. Right? So that vas deferens is going to carry the sperm out of the scrotum, and it's going to carry it into the abdominal pelvic cavity. So right here, we see that vas deferens comes up, and then it comes around right here. On the back side of the urinary bladder, that vas deferens kind of comes up like this. Okay? Now, we're going to take a little bit of a closer look at spermatogenesis and sperm production later on this afternoon. And the sperm in the vas deferens look like sperm. Right? We'll talk about the anatomy of sperm, but I think it's probably one of the more readily identifiable cells of the human body. It's got a very, very unique shape. At this point, those sperm aren't able, they can't swim. Even though they look like sperm, they can't really swim. They can't fertilize an egg at this point. They have to be mixed with the contents of, essentially semen in order to be able to do those things. So they're just kind of sperm being carried by peristalsis through this vas deferens. And then when they get right here, they get mixed with the secretion of these glands that we see right here and right here. These are called the ejaculatory glands, or I'm sorry, they're not. They're called the seminal vesicles. Yeah, seminal glands. Sorry, my brain glitched for a second there. They're called the seminal vesicles or the seminal glands that we see right here and right here. And these seminal vesicles contribute about 60% of the total volume of human semen. The secretion of the seminal vesicle is obviously liquid. It gives a fluid medium for the sperm to be suspended in. But it also is really rich in sugar. That sugar is going to be a source of energy for the sperm as they swim through the female reproductive tract. We'll see on probably Friday how far they have to swim. It's a pretty significant distance, but we'll see how far they have to swim. And then there's also a lot of vitamin C in the secretion of the seminal vesicles. Now, vitamin C is going to act as a pH buffer against the somewhat acidic environment in the female reproductive tract as well. So we're providing energy and protection to the sperm via the secretion of the seminal vesicles. All right. And then once the sperm are mixed, take this thing apart. The sperm are mixed with once they're mixed with the secretion of the seminal vesicles, that fluid travels through this little duct that we see right here. It's called the ejaculatory duct. And it's mixed with the secretion of this gland right here at the base of the urinary bladder. Do you know what gland this is?
Prostate?
This is the prostate gland, yeah. This is the prostate gland. So the prostate gland is, well, a pretty big topic in men's health. We'll talk about that in just a minute. But it also contributes the rest of semen volume as well. I mentioned that these sperm that are here can't really swim and they can't fertilize an egg yet. They can't do either of those things until they're mixed with the enzymes that are in the prostate gland secretions. So once the sperm are mixed with the secretion of the prostate gland, they become what we call capacitated, and they're able to swim through the female reproductive tract, and they also have the ability to fertilize the eggs. Okay? But until that point, they aren't able to do either of those things, even though they look like normal sperm cells. But this is, of course, where the ejaculatory duct ends right here, and then we enter this, which is continuous with the urinary bladder. This is the urethra right here. this is where that anatomy essentially becomes shared between the urinary system and the reproductive system in males. Finish this real quick. So if we put the rest of the penis on there, it's kind of fun to say stuff like that. That would normally be out of context, but for this class, it makes perfect sense, right? So the urethra, of course, leaves the urinary bladder. It travels through the penis, and then it exits the body right here. So basically, everything from here on is completely shared anatomy between the renal system and the reproductive system. We'll talk about the implications of that in just a couple of moments. But the prostate gland is kind of an interesting one. It is found at the base of the bladder. It obviously is contributing important things to semen that will allow the sperm to fertilize the egg. But it also can provide some problems if it becomes enlarged. I think current statistics tell us that 1 in 3 men are going to deal with an enlarged prostate at some point in their lives. Now that could be via prostate cancer. Sometimes it's just a benign enlargement of the prostate that needs to be dealt with. But what can happen is when the prostate enlarges, it can actually pinch the urethra right here, which would make it difficult to start urinating. And it can also impede on the urinary bladder itself, which can also lead to problems with incontinence and problems like that as well. So this is a really common problem that men see as they age, and it is directly tied both to reproductive health and to urinary health. So it's kind of an interesting placement of that gland that we see right here. Okay. To kind of rephrase the purpose of this, right, this essentially makes sperm active. So, when we start to talk about things like in vitro fertilization and stuff like that, eggs, they don't really need to be, more than ovulated before they can be collected. So it's possible with in vitro fertilization to go in and actually collect eggs almost directly from the ovary. We can't go into the testes directly and collect viable sperm because those sperm have to be mixed with the secretions of the prostate. They essentially have to be ejaculated in order to be viable, right? So that's kind of a big difference between the viability of male and female reproductive tracts, is that these sperm have to be ejaculated to be viable because they have to be mixed with the secretion of the prostate. So the prostate is divided into different regions. We see here that this section, as it travels through the urethra is divided into different regions. As it travels through the prostate, we call this the prostatic urethra. And we have the membranous urethra, and then as it travels through the penis, it becomes the penile or the spongy urethra. So it's divided into regions depending on where we're at. That name, spongy urethra, comes from the anatomy of the penis itself. The penis, of course, is the copulatory organ. Its job is to essentially deposit semen into the female reproductive tract. So it's made up primarily of erectile tissue. That erectile tissue exists in a couple of main areas. We have an erectile body right here. This is called the corpus cavernosum, which literally means cavernous body. And then we have the corpus spongiosum, which directly surrounds the urethra. If you put this model together, you can see those chambers a little bit more clearly. Right, so if we look here, you'll notice that you have the corpus spongiosum, or the spongy body, directly surrounding the urethra right here. And then you have those corpora cavernosa here and here that are responsible for also filling with blood during erection. Also, this model, it makes a cute little face, right? So what happens is, this is really, it's not completely hollow, but it's a hollow-ish space. And so so what happens is during sexual stimulation, the blood vessels that supply the penis with blood dilate because of parasympathetic nervous system stimulation. And so there's a massive amount of blood flowing to the penis. And as a result, the blood vessels that are draining the penis actually get pinched from all of the blood flowing into it. So blood can get in, but it can't get out. And as a result, all of this erectile tissue fills with blood, and the penis becomes erect as a result of that. Probably didn't need a demonstration. So anyway, it's kind of an interesting way in which this occurs, but this is just sort of passive filling with blood, and it becomes erect as a result. So, what else was I going to say here? Also, so we have the seminal vesicles, or the seminal glands. We have the prostate gland right here. We have a third gland as well, and this model shows it particularly well. This little gland right down here is called the bulbourethral gland. The bulbourethral gland is, it doesn't contribute to semen volume. That's the job of the seminal vesicle and the prostate gland. But because this is essentially shared anatomy with the urinary tract, and sperm are pretty delicate cells, they're really sensitive to their environment. What we need to do is before ejaculation, we need to clean the urethra of any residual urine and things like that. So the bulbourethral gland actually, its secretion cleans the urethra prior to ejaculation so that it helps to increase the viability of sperm. Right? Let's see here. So what else do we have? So the penis is fairly straightforward, I suppose. Obviously, the skin has been removed here because of the way this model was dissected. But we do have the glands of the penis right here. Now, if this were intact, the glands would have a piece of skin going over it like this. It's called the prepuce of the penis. It's kind of what we colloquially call the foreskin of the penis. And that would be removed during circumcision, which is a really common medical procedure, at least in Western medicine. So what we would see then is in a circumcised penis is just the glands of the penis right here. That foreskin or the prepuce would have been. So yeah, so we have that. I think there was something else I wanted to say about this. But it's a fairly straightforward set of ducts. But this one actually does. It's got a nifty little magnet that you can just lift off and see the inside of the testes. Isn't that cool? Look at that. Astounding. So when we take a look then at the testis, we can see that, as we mentioned this morning, you know, the inside of the testes is made up largely of these seminiferous tubules. That's what they're, I think, trying to demonstrate with these squiggly little lines, right? Are all of these seminiferous tubules here and here and stuff. You can see that they're organized pretty well into lobules. There's a lot of white connective tissue between them. That's that tunica albuginea that you guys are asked to identify in your lab. All of those tubes converge on this structure. This is called the rete testis, and then ultimately into the epididymis, which comes down here. Whoops, sorry. Which comes down here, and it makes a U-turn, and then it goes. What we want to do is we want to go ahead and take a look at this part of the testis. And we want to go ahead and kind of correlate what we're looking at under the microscope with what is happening in terms of spermatogenesis. So let me come back over to PowerPoint really quick, and we can go ahead and do that. I'm going to go ahead and kind of break it down a little bit to make it palatable. So remember, remember that when we look at gamete production, we're looking at specifically. Because I'm actually running a lecture, I'm not going to make you guys relearn the steps of analysis or anything like that. You're not going to need to tell me on the test what happens during anaphase 2 or anything like that, because you probably don't remember, and honestly, I'm not entirely sure I remember either. So that's okay. I'd have to look it up. It's been a minute. But I do want us to have some primary takeaways from meiosis, right? Remember that meiosis is different from mitosis in a couple of primary ways. Mitosis, the goal is for a cell to create an identical copy of itself, right? Meiosis, the goal is to produce gametes, to produce haploid cells, and from a diploid, from the diploid parent cell that we start with, that enters meiosis, how many haploid daughter cells do we get? Remember? Should be 4, right? Okay, should be 4. Because we do go through the process twice, we should end up with 4 cells instead of 2 as a result. Right? So again, I'm not going to make you relearn all of the steps, but that is an important feature. Spermatogenesis, the process of sperm formation, is about as textbook meiosis as you can get. So when we start to look on Friday at what we get, the process of egg production, we're going to see that there aren't necessarily rules broken, but there are definitely things that we need to consider as we take a look at it. It's not as textbook. But for spermatogenesis, it's like textbook meiosis, which is really, really helpful. So the first takeaway is that we're getting 4 daughter cells, so 4 sperm cells from our starting cell. Okay, we'll talk about that in just a couple of minutes. The other takeaway is that those daughter cells, those sperm cells that we're producing, they have half of the DNA of the starting cell. Now, is the DNA in those cells only the maternal or only the paternal DNA? So in other words, are you only passing on your mom's or your dad's DNA to your offspring? No, we're passing over. We're passing on a combination of mom's and dad's DNA, right? And there's a process you guys might remember from meiosis called crossing over, in which those maternal and paternal chromosomes exchange certain genes or sections of DNA with each other, right? So again, we're not going to look at every phase necessarily, but I do want you to understand that the gametes are not just paternal and not just maternal DNA, but instead it's a combination, and that's really important. In the same way that sexual reproduction increases genetic diversity, we see that it wouldn't be the same if we didn't have this crossing over during meiosis. Okay, so with that said, let's go ahead and take a look at this process. So the cells that we start off with, the cells that we're born with, are called spermatogonia. You can see those right here. These spermatogonia are essentially diploid stem cells that have the potential to become sperm. Right? We're going to have the chance to look at spermatogonia under the microscope in just a little while. But spermatogonia are these big round cells with big round nuclei. And they're diploid. Just like any other cell in your body, these spermatogonia are diploid cells. They contain a maternal set of DNA, they contain a paternal set of DNA. This diagram tries to show that by just showing a single chromosome of 2 different colors. So here it's showing a blue and a red to indicate that it's a diploid cell. It's got maternal and paternal DNA. The other thing that this diagram does is it separates out what happens during embryonic development, and then as we move through, what happens after puberty starts. And so we can see here that these spermatogonia are present in embryonic testes, and they are undergoing mitosis throughout embryonic development. Okay? And then when we're born, we have these spermatogonia in the seminiferous tubules of the testes. And they just kind of sit there for roughly 10 to 13 years until puberty begins. And then we start this kind of reproductive phase of our life once puberty starts. So these spermatogonia do undergo mitosis, right? They do undergo mitosis. And when they undergo mitosis, they do what any cell that undergoes mitosis does. They produce identical copies of themselves. Right, so we're producing identical copies of these cells right here like this. We can see that happening in the upper part of the diagram that I projected as well. It's just simply a mitotic. Now, when puberty begins and we start to make sperm, spermatogenesis begins. When puberty and spermatogenesis begin, we see kind of an interesting thing occurring. We see that of these two daughter cells, only one of them is going to enter meiosis. Okay. Only one of them will enter meiosis. The other one is going to hang out in the seminiferous tubule and continue to undergo mitosis. So because of that, what we see happening is we see that we don't really ever run out of these spermatogonia because only one of those two enters spermatogenesis, enters meiosis. The other one stays in the seminiferous tubule to continue to undergo mitosis. In theory, males can produce sperm for the rest of their life. And they produce about 200 million sperm a day. Okay, so we're talking about a huge process here, about 200 million sperm a day that are being produced. Once puberty starts, theoretically for the rest of their life. And the reason that they're able to do this and never ever run out is because one of those daughter cells is going to continue this mitotic process, and the other one is going to enter meiosis. When we look at oogenesis, the production of eggs, in a couple of days, we're going to see that that's a starkly different process. Okay, so we'll take a look at the production of eggs on Friday, and we'll see that it's very, very different for them. But for sperm, we maintain this constant stock, if you will, of spermatogonia in the seminiferous tubules and the testes, so that they're able to survive, okay, and continue to make sperm. So what we end up with then is we end up with this one that enters meiosis. We end up with 1, 2, 3, 4 spermatids. Okay. Excuse me. We end up with 1, 2, 3, 4 spermatids. So, in addition to these being haploid cells that contain half of the amount of DNA as the daughter cell or the stem cell does, what we also see is that they undergo a significant change in their shape, right? The morphological differences between that spermatogonium that we started with and the spermatids are significant, right? Not only in terms of the overall size, but in terms of the shape. We're going to see, when we look at these under the microscope, that the spermatogonia are big, round cells, and then those spermatids are these really streamlined cells that, again, have this really unique shape that's probably the most recognizable cell that humans actually make. Right? So, all right, come back up here. All of this is happening in the seminiferous tubules. Right? So if we've got these seminiferous tubules in the testes, just oodles and oodles of them. We're looking at the cross section here. We've got all of these seminiferous tubules here. Right? The micrograph that I have projected on the board right now is just a section of one of those. So what we're looking at here under the microscope would be like a section of one of these seminiferous tubules. So it would be something along the lines of like this. So the lumen of the seminiferous tubule is right here. This is the lumen of the seminiferous tubule. This is where sperm would eventually find their way to and be carried away to the epididymis and the vas deferens. So we're looking at multiple cell layers here. Notice the basal layer of the seminiferous tubule. Notice that basal layer, you've got those big round spermatogonia that I was telling you about, right? Big nucleus. So those spermatogonia that do intermeiosis, what they do is they start to migrate towards the lumen of the seminiferous tubule. And as they migrate towards the lumen of the seminiferous tubule, they're undergoing meiosis. By the time they're ejected into the lumen of the seminiferous tubule, we see that those cells have undergone a significant change in their morphology, right, their shape. Those nuclei are significantly smaller and they're much more streamlined. And you might actually even see sperm in the lumen of your seminiferous tubules today. We're going to look at sperm under the microscope anyway, but you might see them on this first slide. So one of the things I would like to look at under the microscope is the seminiferous tubule in cross-section so that we can take a look at that. So if you guys would do me a favor, there's a slide back here in the and it's also a lot narrower. It's not ball-shaped. It's just a little bit more narrow. But we want to be over here on the testis itself. So really what we want to do then, once you are sure you're on the testis, and I'm more than happy to come around and help, is we just want to zoom in and we want to take a look at what we have here. So I'm going to zoom in. We try to find a seminiferous tubule that looks good. Some of these look great, actually. Some light. We'll take a look at what we have. All right, so now I'm pretty zoomed in on these. Let me focus it a little bit. There we go. So I'm pretty zoomed in on these. So we have a seminiferous tubule right here. We have another one right here. We have another seminiferous tubule right here as well. So again, just find one that looks good to you. This one looks particularly good. What we see, of course, are those spermatogonia very close to the basement membrane. Again, we're looking at big, round nuclei. We're looking at big cells. But then you'll notice that by the time those cells get to the lumen of that seminiferous tubule, heck, there's an entire sperm right there, right? It's all these little hair-like things. Those are all the tails of sperm that are being ejected into the lumen of the seminiferous tubule. So as we go from here to here, those cells are going through their meiotic divisions, and we're ending up with these haploid gametes that are being kicked out into the lumen of the seminiferous tubule. Even though we produce about 200 million sperm a day, this is not a fast journey. From spermatogonium, from diploid spermatogonium to haploid gamete is about 2 to 2.5 months, is about how long that process takes. So the meiotic divisions take about 2 or 2.5 months. Okay? So let's first of all, make sure you're on the testis and not the epididymis, and I can help you with that. And then once you are sure that you're in the right spot, take a look, find a seminiferous tubule, identify those spermatogonia, and let's try to see if we can see little spermatids in there as well.